Okay, welcome back, ladies and gentlemen. Thank you all for joining us again this morning. We're gonna pretty much jump right into it and crack on with the day. So first of all, obviously I know there's a couple of new faces here from yesterday. Thank you again for joining us. You're in the right spot. We've got there's going to be a mid-morning break, I should say, at 10:35 to 11. So there'll be coffees, refreshments, and things outside in the Wilson Suite where the exhibitors are. And then lunch again is same place as yesterday, but it's 12:35 to 1:45 in Loria, which is the restaurant area on this level, just around the corner over the Koi Pond Bridge. And we will be finishing a little bit earlier than planned today as well. I know we moved Ryan's talk forward at the end of the day yesterday in lieu of Vivek Gupta, who unfortunately was unable to join us. So our goal is to share Vivek's session virtually with all the on-demand talks after the summit, where we post these on our site. So we'll be finishing around 3:15 p.m. today, give or take, maybe a little bit before that, depending on the speed of the talks. But just a heads up on that. Obviously, I'll send a reminder to you all and keep you posted on that. We're going to jump right into it this morning again, and I'm going to hand things over very briefly to Firuz. He's obviously going to say a couple of words as our chair for the day, Master of Ceremonies, and then obviously he'll be going ahead and introducing speakers. There will be a little bit of time at the end of each session for Q&A as well, so definitely continue the questions where appropriate. And without further ado, Firuz, over to you.
Again, thank you everyone for being with us today. The talks today will continue on the predictive tools that as we had yesterday, but also solubility enhancement, additional topic that is going to be presented today, plus biological deliveries, as you have seen from the agenda. Our next speaker is Mikhail Murashov. Yes. And he is a senior scientist at Sterile and Specialty Products at Merck. Today, I will not read through the bio. I will let everyone to talk more about their bio as needed based on what they believe appropriate. But Misha is going to talk about predictive power of SCISSOR technology from Pion in subcutaneous injection at site. Which allows, obviously, reduce the number of animal studies, hopefully, and predict what's going to happen in vivo. Again, thank you so much for being with us.
All right, hello everyone. Can everyone hear me okay? All right. My name is Mikhail Murashov. I go by Misha. I'm an associate principal scientist in Biopharm Group at Merck. Simon, thank you. Firuz, thank you, and our organization committee for this opportunity to present. So today I'm going to give a talk on the predictive potential of power and operational use of SCISSOR, or subcutaneous injection site simulator, to streamline subcutaneous biologic drug development. And I actually gave another talk 2 years ago in Philly in the same summit on SCISSOR on a literature analysis for the predictive power of this. So this is more like a sequel to the talk that I gave 2 years ago. All right, so for the objective for this talk, and as majority of you are from pharmaceutical industries, you probably already aware of this, but predicting bioperformance, specifically bioavailability of subcutaneous administered biologics, remains a large knowledge gap in drug development everywhere. Underlying causes for this clinical bioavailability spread of commercial monoclonal antibodies, which is between 40 to 90%, is very unclear and not entirely understood. Furthermore, there's no established reliable translation from preclinical species to human or reliable in vitro method that predicts clinical performance. This lack of understanding creates uncertainty in formulation development, clinical dose estimation, and supporting IV to subcutaneous formulation transition later in the development. Thus, there's an urgent need for development in the biopharmaceutics performance prediction models in vitro, in vivo, or in silico. So in this presentation, we will actually discuss the predictive potential or power and operational use of one of the in vitro models of interest, and it's called SCISSOR, or Subcutaneous Injection Site Simulator, that was developed originally by University of Bath in UK. Then it became Sirius, and now it's Pion. And please correct me if I'm wrong, but currently it's owned by Pion and it's commercially available. So in this schematic, it's pretty straightforward, where it shows that currently we don't really have a good IV-IVR or in vitro in vivo relationship from even in vivo models to the clinical performance when it comes to subcutaneous biologics. And for the remainder of the talk, I will be primarily focusing on subcutaneous biologics, but specifically subcutaneous monoclonal antibodies and fusion proteins. So the idea here is that we would like to utilize SCISSOR to bridge this gap or give us some clue of how our formulations will be performing in the clinic. So what is Pion SCISSOR, or what is Subcutaneous Injection Site Simulator? So it's an in vitro model for investigating the performance of subcutaneous formulations, small molecules or biologics under simulated physiological conditions. And when I say simulated physiological conditions, they try to mimic your physiological conditions of your subcutaneous tissue as close as possible. So the pH, the temperature, even some of the other things that are in your subcutaneous tissue are tried to be mimicked on a basic level, but they are mimicking this in their system. And the abilities of the SCISSOR are as depicted. You're able to inject formulations into this extracellular matrix that is commercially available from Pion using hypodermic needle. You're able to inject between 0.2 to 1 ml, which is clinically relevant because a majority of the subcutaneous biologics formulations volume of injection is around there. You're able to measure and monitor the pH at the injection site, as well as the pH at the sink chamber, as well as monitoring the temperature as well. So like I said, it tries to mimic the pH conditions, the temperature conditions of your subcutaneous tissue. We able to use 50 to 300 ml chambers, so ultimately it's just a sink chamber into which your formulation will be diffusing to. And the difference, honestly, if you are using a very low concentrated formulation, the 50 ml chamber would be preferable because then the concentration will be higher, so easier for detection. The next thing is the monitor transmission through the cartridge. So there is 4 LED channels that shine through your cartridge and able to give you a transmission data through it. And the idea behind this is that it's able to give you a clue if there's any precipitation, aggregation, or gelation events happening in your injection site after you injected your formulation. With the current system, which is actually depicted as your top right corner, that's a SCISSOR N3 system. It actually has clear chambers, so you actually visually see what's happening with your naked eye, as well as there's cameras that actually depict anything that's happening to your formulation within the ACM. In the previous system, which was SCISSOR N1, the chamber was closed off, so it was very difficult to see, so we were relying heavily on that transmission data. And finally, the most important bit, being in biopharm, is ability to remove the samples from the sink chamber for quantitation at different time points, which ultimately gives us the % drug release profile. So that's something that we're most interested in, because that's the data that we would like to see if there's any correlation between that release profile to our clinical performance on when we screen formulation candidates. So in the case study 1, we're going to go over what our process of thinking was when we got this instrument and when we read all the literature. And again, I would encourage you guys, if you're interested in this, I'm sure the presentation is still on demand from 2 years ago when I went over the literature analysis on the predictive power of this instrument. But ultimately, in a short summary, there were some publication that said that there is a correlation, and some publication said that there's no correlation between the release profiles and the in vivo data, or in clinical data. So we wanted to, test the instrument ourselves and see what we can get out of it and see what kind of data we can get out of it for our pipeline support. And ultimately, we started with this case study 1, which was the parameterization for formulation head-to-head comparison. Ultimately, we wanted to know what parameters we need to keep consistent in order to have a meaningful head-to-head comparison between your formulation candidates. So initially, we conducted this evaluation of different formulation of this monoclonal antibody 1. This was in SCISSOR N1 system, so there is no, not a lot of runs, so therefore there is no standard deviations and errors in these graphs. The purpose was to determine if SCISSOR could distinguish between the various formulations while keeping the injection volume and formulation concentration consistent. So ultimately, first we wanted to know if we run different types of formulation of the same monoclonal antibody, will it be able to differentiate among them? And then, other than that, we wanted to evaluate the effect of the injection volume and evaluate the effect of the formulation concentration on the release. So as a result, SCISSOR successfully differentiated among the different monoclonal antibody 1 formulations as predicted, which is depicted in figure 1A, and accurately depicted the fate of the formulation at the injection site, which is data not shown here, but we will discuss this in upcoming slides. And assessment of the effects of the injection volume, which is in figure 1B, and concentration in figure 1C on the release profiles revealed that only the volume of injection affected the release profile in SCISSOR. This was very important because now we knew that if we want to evaluate, formulation candidates in this instrument, we have to keep the injection volume consistent. So for the case study 2, we took another monoclonal antibody, which is in this case monoclonal antibody 2, and we conducted, again, SCISSOR N1 work with this, which, with this monoclonal antibody that underwent formulation changes ranging from minor, such as an excipient change, as well as major in the process change. So this was a good example because here we wanted we would get a lot of questions, is that if, the formulation change in excipient, what was the risk associated with it? What's the PK risk associated with it? Or what if the formulation, undergoes a major change, such as in this monoclonal antibody case, the process by which the protein gets derived changes. So we get these questions a lot, so that's why we would like to use this SCISSOR to see if it's helpful in guiding us or giving us clues, in addition to the other experiments and initial work that we do. So in the case of the process 1 versus process 2 formulations for this monoclonal antibody, SCISSOR yielded comparable release profiles with minor difference and extended the release at 24 hours, which is depicted in figure 2A, which was actually confirmed by preclinical studies, so that was good. And then in the formulation, additionally, in the formulation surfactant screening for monoclonal antibody 2, SCISSOR successfully determined the necessity of the surfactant in the formulation and demonstrated the comparable release profiles between poloxamine 188 and polysorbate 80 containing formulations, which is depicted in figure 2B, which actually further aligned with the results from our analytical comparability. And yes, this is maybe one example and whatnot, so I cannot really, extend this to all of the formulations, but in this case, for this example, it actually aligned with other results and it was pretty helpful. So as you guys can see here, again, I apologize, there's no standard deviations because the SCISSOR N1 system, we were limited at the runs that we could do, but ultimately, we see that, in different process 1 versus process 2, the release profile is pretty comparable, and then there is change in the excipient, such as in this case surfactant, which is poloxamine 188 versus PS80, we get comparable results. However, we also see that the surfactant is needed in the formulation because we see that if we run the formulation without a surfactant, we actually get a lower release, extent of the release, as well as just a underperforming release profile for that formulation. So after those 2 case studies, after those experiments, we wanted to summarize what formulation changes can or cannot be assessed in SCISSOR. And as simple as this table is, this is probably the most asked information of us by different other departments, such as device or formulation or any other groups, is that they all want to know what kind of things we can evaluate with this instrument. So, like I said, as simple as this table is actually one of the most informative table when we talk about this instrument. So the changes that we can assess in SCISSOR are depicted here in green, such as change in the injection volume, change in the formulation composition, introduction of a novel excipient, change in the formulation attributes, such as pH, osmolarity, viscosity, etc. However, the following things cannot currently be assessed in SCISSOR, such as change in number of injections, change in device, anatomical site of the injection assessment, or depth of the injection, which is inadvertent IM injection de-risking. So next, we, after running those experiments, as you noticed, a lot of those release profiles don't reach 100%. So we wanted to know what happens to the rest of the protein. And in addition to that, we also noticed that if we run highly concentrated MABs, in this case, we depict MAB1, we noticed that there was what we refer to as cloudiness at the bottom of the cartridge at the end of the experiments at 24 hours. We hypothesized there was protein at first, but when we connected with Pion and everyone else and tried to read through the literature, none of them really characterized what that cloudiness was. So we kept referring it to as cloudiness, but we couldn't really call it protein yet until we figure out if it was protein or not. So in this case, we connected with the analytical team as a collaboration, and I'm not an expert in this field, but I will try to cover this as much as possible. We provided them cartridge at the end of the experiment for monoclonal antibody 1, and we provided them a cartridge with the placebo experiment of monoclonal antibody 1, but obviously without protein. And we split that cartridge into top, middle, and bottom fractions and send it for the analysis. And the analysis is depicted in the slide, which is for concentration turbidity, size exclusion chromatography, dynamic light scattering, and AUC. And the cloudiness that I'm referring to, you guys can see on the monoclonal antibody 1 cartridge at the bottom. Oh. Right here. You guys can see it right there. And maybe another note on this is that when we went back and talked to Pion about this and we looked through their case studies that they used for educational purposes, their monoclonal antibody case study that they used actually experienced the same thing. And when we talked to them about this, there was actually a common phenomenon. So it wasn't only relevant to our formulation per se. So any highly concentrated monoclonal antibody that we ran, we actually see this precipitation or cloudiness happening at the end of the experiment. So the first thing that, as simple as it was, but very important we were interested in is the mass balance. And the idea is that for this monoclonal antibody 1, going back to case study 1 and case study 2, or case study 1 in this case, we saw that at 24 hours we get around 74% drug release. So we wanted to account for the remainder of it using this analysis of what was left in the cartridge. And to our pleasant surprise, we actually were pretty spot on by evaluating top, middle, and bottom fraction for the monoclonal antibody 1 formulation, and we achieved around 26.2% protein that was left in the cartridge. So the mass balance worked out pretty well in this example. And then based on the turbidity, we also see that majority of the protein is located in the middle to the bottom fraction of the cartridge, which is giving us a clue that the cloudiness that we see is related to the protein. So for the conclusion here is the total leftover protein that were estimated by the protein concentration reveals that about 25% of the original total loading of monoclonal antibody 1 remains in the cartridge at 24-hour time point. And top fraction has the least protein, where middle and bottom fraction have much higher protein amount. Then we did size exclusion chromatography, and here the idea was to see if the aggregation of this protein accumulation is protein related, or because we run placebo versus monoclonal antibody 1 control, and we see that for the placebo control, we do not see any peaks in the UPSAC. Where for the monoclonal antibody 1, we see this nice peak, which tells us that the aggregation that we're seeing is protein related. In addition to that, we see that, looking at different top, middle, and bottom fractions, we see that the bottom fraction has the most protein in it that actually has aggregation and fragmentation events that we're able to capture. So as a result here, the top, middle, and bottom fractions remaining in the CM cartridge after monoclonal antibody 1 release clearly show difference in the amount of protein and aggregate formation. And protein aggregation is detected in the middle and bottom fraction with increased trending towards bottom fraction. Next, we looked at dynamic light scattering, and ultimately here we again looked at the top, middle, and bottom fraction, and the result here was that the size distribution of analytes in each fraction confirms that the presence of larger sized aggregates in the middle or bottom fractions of the ECM cartridge. So we see that majority of our fractions are in the middle and bottom, which is further aligning with the result that our protein is located towards the bottom of the cartridge. And then finally, for the analytical ultra-certification, here, as a result, what we saw is that the enhanced separation for low molecular weight and high molecular weight species based on their sedimentation event, and that the higher molecular species indicative of protein aggregation, again, observed in middle and bottom fraction with increased trending towards bottom fraction. So ultimately, all these results point to the same conclusion, that the cloudiness that we see is due to the protein aggregation, and it has nothing to do with the ECM degrading or anything else. It actually is due to your formulation. So then we move to the case study number 3, which is one of the, informative slides here on the biopredictive potential. So last year, we had a summer intern working with us, and we decided to do this project where we took 2 commercial monoclonal antibodies that was very low in the clinical performance, in this case Kunthium, or in the US it's known as Silic, which has around 55% clinical bioavailability. And then we took one of the best performing ones currently commercially available, which is Praluent, which has around 85% bioavailability. And then we took monoclonal antibody 1, which falls somewhere in the middle. And we wanted to know if SCISSOR is able to differentiate among them, number 1, and able to rank order them based on their clinical performance. Here, we're not necessarily looking for that correlation yet. We honestly just want to know, knowing their clinical performance, if SCISSOR is able to appropriately rank order them based on known clinical performance. So is the result, as you guys can see here in figure 3A and figure 3B, indicated that SCISSOR effectively and significantly ranked these 3 MABs utilizing the release data up to 6 hours, % drug release and area under the curve with statistical significance. So ultimately, it was able to differentiate among these 3 MABs according to their clinical performance. However, the resolution was only seen up to 6 hours. And it actually followed pretty well with the known literature about SCISSOR and ECM. And for some of you that might not be familiar with this, is that the War publication came out that ECM, is hyaluronic acid based, number 1, and that hyaluronic acid starts to degrade around 6 hours. So in the earlier publications for SCISSOR, they all used diffused fraction at 6 hours. We do 24 hours because this is the maximal time to run this experiment. So we use the data to the full, but then we decided to see if we can segment it and see if we can maybe derive something looking at 6 hours alone. And again, if you look at the figures B, C, D, and E, B is the % drug released at 24 hours, C is the AUC to 24 hours, and we see that looking at those two things, we do not see significant differences among these three MABs because at 24 hours, everything kind of goes to the same point on the extent of the release. However, if you look at 6 hours, percent drug release and AUC, we actually see significant difference and separation and differentiation among these MABs based on their clinical performance. And the result here was surprisingly, pleasantly surprising for us because this, shows that it's able to differentiate this. The resolution is not necessarily there because here we use extremes. We use the worst, the best, and somewhere in the middle. However, I do know that Pioneer is currently working on improving their ECMs that's supposed to improve this resolution, and you're able to run experiments for longer. So hopefully with their improved ECM, we're able to get a bit of better resolution and see the differences not only on the extremes, but maybe two MABs that are somewhere in the middle, and you still be able to see the difference. So as a conclusions, SCISSOR system is able to capture the behavior of the formulation at the injection sites. It detects any precipitation, aggregation, or formulation at simulated site of the injection via transmission imaging data. It has the capacity to differentiate formulation with different release profiles, which is valuable for excipient or formulation screening and formulation development. Has the potential to obtain this in vitro in vivo relationship or IVIVR based on their release profiles in preclinical and clinical assessment. However, the translatability of the SCISSOR results and its predictive power to the clinic has not been fully demonstrated as supported by multiple publications in the literature and our internal analysis. So the predictive power or potential of SCISSOR needs to be investigated further with more examples of different types of subcutaneous biologic products and their corresponding clinical data. To establish this IVIVR, SCISSOR could be routinely incorporated in the subcutaneous biologic drug development workflows. So then for the acknowledgement reference, I would like to acknowledge some of the direct contributors at Merck, which is Mikhail, Susan, Aparna, and Yash. And for the references, if you are all interested in looking into all of the SCISSOR-related publications, they're depicted here. They were also used for that presentation I mentioned to you guys 2 years ago on the literature analysis as well. And with that, again, thank you so much for your attention. I'll open the floor to questions.
Questions?
Thank you. That was a really nice talk. It seems like there's been a lot of progress in this area in the past few years. I was curious with the data that you showed with the aggregation and the protein settling to the bottom of the cuvette. If I recall, these cuvettes have a little stir bar inside of them. And have you looked at or seen any correlation between that either aggregation event or the settling that you are seeing occur as a function of the stir speed?
It's a great question. We did. So your precipitation happens regardless if the stir bar is there or not. In fact, I think Pine is currently thinking of removing the stir bar altogether because it seems to be not really making any effect on what's happening. We did run experiments without stirring and even without stir bar. And we do see this precipitation, however, it's not as spread out at the bottom because it's kind of settles down at the bottom, but you still see it there.
Any more questions for Isha before we move on?
So Merck has Merck ever used this equipment for small molecules?
Good question. So my department and where I'm at in the sterile and specialty products at Merck, we currently using it or focusing to use it on biologics, so primarily medical antibodies and fusion proteins. However, we do have another SCISSOR at Merck in animal health, and they use it on small molecules.
Okay.
So, but my own experience is primarily with biologics with that instrument, but it is meant for small molecules as well. We just don't have experience. Don't have any experience with it.
Okay, and do you have the capability to change the pore size of the cartridge?
Good question. So by standard, by default, it's a polycarbonate membrane, if my memory serves me right, and it's a 5-micron pore size. You can get custom membrane. You can get either methyl cellulose or something else, but 0.2 microns. But please keep in mind, the membrane has no interaction with your protein. It does not make an effect on your release. So the idea behind SCISSOR is that all of your interactions between your formulation and ECM has to happen between your formulation and ECM. And then once ECM releases the protein, the membrane should not interfere with its release to the sink condition. So what I'm trying to say is that those membranes technically act as fence to keep ECM in the cartridge, but should not have any effect on your release. And if it does, you need to change the membrane apparently. Yep.
Thank you.
Any other questions? Thank you.
Thank you, everyone.
Thank you.